First, I regret to say that I possibly cannot contribute much to the question of creatinine and biomarkers and dexpramipexole, although I find this issue very interesting. I rather give you a view of the future, and you will hear many ifs if I refer to biomarkers. But perhaps it gives you an idea, a different view. Secondly, those who attended the WFN meeting, there are not many, so it's not a big problem. I borrowed, of course, 6 or 7 slides from Johannes Brettschneider, who did the work on the staging, and therefore you perhaps get a little bored, but I try to make it interesting instead. Where do we start from? In my view, I think we accept now that the staging of ALS involves a continuous process which starts focally and spreads in most patients, perhaps everyone, but at least in most patients, continuously. In this patient, the disease started at the right hand, and then he tells us the second part of the body that was affected is the right leg. And I guess most experienced ALS clinicians will tell you which is next. It's the left arm. And of course, this is a phenomenon which I was aware of but did not invest much thought to in my career as a neurologist, but many others did as well. And I think John Ravitz's ideas opened our eyes, and I like this idea, and I tell my residents that this is the way the stages of ALS should be defined. What is seen? How can we explain this? We only could explain this by analogy. We could explain it by the work, the longitudinal neuropathological work as done by the Braques in, for example, Alzheimer's. The tau pathology spreads from the hippocampus into other parts of the limbic system and the temporal cortex, and then the entire neocortex, and more spectacularly even for synuclein. And he showed us 10 or 15 years ago that synuclein, a molecular marker protein of Parkinson's, of course, spreads from the periphery, from the olfactory bulb and the gastrointestinal tract, cranially through the brainstem into the substantia nigra, and then into the neocortex. Why could he find that? Because he studied the disease longitudinally, and he had a new method, or a method which is not often employed. He uses thick sections. In the beginning, 100 micrometer sections. In the previous years, he discovered that he could do this also with 70 micrometer sections, and even the non-pathologist sees immediately that with using these thick sections, you see much more pathology. For example, here the structure of the hippocampus in comparison with the conventional sections, much more clearly. And he and Professor Brettschneider used this approach for a staging of ALS. And they defined 4 stages of TDP-43 pathology. This is now TDP-43 and not synuclein. And in the first stage, they found the TDP-43 pathology, and perhaps not surprisingly, in the motor cortex, in the bulbar motor nuclei, and the spinal cord motor neurons. It is interesting to note here that they used a marker for phosphorylated TDP-43, phosphorylated TDP in the TDP-43 in the somatodendritic compartment of bed cells and anterior cells alike, but they only found phosphorylated TDP-43 in the peripheral axons of the motor neurons, but not in the corticospinal tract. I will come back to that. The second stage is characterized by more widespread pathology of the frontal cortex and involvement of the infraolives or the other precerebellar nuclei. The third stage is characterized by that the TDP-43 spreads into the entire frontal cortex and affects also striatal neurons. And the fourth stage is characterized for a clinician, very surprisingly, by hippocampal TDP-43 pathology and even cell loss in this part of the brain. So there were many surprises found by this approach. First, this was no surprise that the ocular motor nuclei were spared by the TDP-43 pathology, but we have to conclude that the term motor neuron disease, many speculated on that and saw this in the past, might be not very correct, because many other, and I don't read it to you, other neurons are affected by this pathology in the thalamus, in the dentate gyrus, and I also told you already that it's even in the Ammons horn. It is interesting to see that the vulnerable neurons show strong and direct monosynaptic cortical input, and this is perhaps the favorite drawing of Johannes Brettschneider. It's not from me. He thinks, or he suggests, that he has seen a spreading of the cortical pathology, and during this spreading, one monosynaptic tract after the other is affected, and this spreading along this afferent pathways makes or induces involvement of subcortical structures. Neurons without direct cortical input, this is the other argument, are spared. For example, the locus coeruleus, which are involved in synuclein pathology, and also, as Brag has shown 2 years ago, very early on in the tau pathology. So this is what I showed you before. The disease spreads along the entire cortex and then affects all the monosynaptic tracts, one after the others, with a few exceptions, I have to admit. So what does this mean? Of course, it is interesting to look at this principle, that it is maybe also true for ALS, like in Parkinson's and Alzheimer's, and what is the basis of spreading and how to interfere with spreading, but we talk about biomarkers. So the first question is, if we want to use this as a support for development of the biomarkers, does this neuropathology translate into clinics? I mean, the first view is, of course, to translate it by imaging methods into clinics. I come back to that. Perhaps we can support these imaging markers, for example, by tract imaging, I come back to that, by examinations of neuropsychology and ocular movements. And of course, the final goal would be that if we use stages of ALS, we could reduce the noise from our biomarker studies. We measure biomarkers in initial studies, late studies, medium studies. We don't know what we do, to be a little provocative. So the question is, can we translate these stages into living patients with ALS? And I give you one example, how we cannot do that. Many groups, and our group as well, try to measure TDP-43 in biofluids. I give you two examples. We can give you another equivocal example by our study, and you see that there is a huge overlap between controls and patients, and also this study looks a little bit more convincing, CSF study, but I think one has to be reluctant to accept it, and the same is true for plasma levels. So in this regard, I think we can learn a little bit where the mistakes or the wrong strategies may be. If we accept the stages of TDP-43 neuropathology, intracellular and synaptic TDP-43 levels are much more of interest than the extracellular levels which we measure. Therefore, this might explain why we cannot reproduce studies, or they show a large variation, and that there is a considerable overlap between ALS and controls. That it also might imply that longitudinal, in this, for studying of TDP-43, longitudinal CSF studies may be more important than blood studies because we only know about TDP-43 pathology in the central nervous system, at least now. And I also mentioned in the beginning that there is an interesting difference between the accumulation between phosphorylated TDP-43 and non-phosphorylated TDP-3 in the respective tracts. So how could we do that? How could we translate this staging into humans? I discussed with Adriano already, TDP-43 this morning. We would like to have a TDP-43 PET. Of course, this is a little bit far away from now. And the other way to address this, we could assess the TDP-43 pathology through a backdoor by associated neuronal loss. It has been shown by 4 studies that the extent of TDP-43 pathology, at least roughly, more than roughly, correlates with the extent of neuronal loss in 4 studies. And therefore, I think it is a way to address these issues to use volumetrics like Turner proposed, or tract imaging to translate it into clinics. This is an example. We prefer tract imaging. This is an example to characterize these tracts by DTI. The corticospinal tract is not a difficulty, but if you look at the corticorubral or the corticostriatal, you better seek advice by an experienced neuroanatomist. It's not that easy. I can tell you, we tried that, and it is partially successful at that time, but this is the way we propose. This tract imaging as a biomarker for the TDP-43 staging system should be, of course, supported by neuropsychological measures of neuropsychological impairment and potentially oculomotor changes. It would reflect the frontal impairment and the impairment of the olivary nucleus. And then, if we are rigid, we could do traditional but stage-dependent biomarker research. It's an if. I told you there are many ifs. Oh? No, it doesn't work anymore. No. So the second steps should be, at least if we learn from TDP-43, we have to compare, at least if we want to represent with our biomarkers the CNS pathology, we have to compare CFF and systemic biomarkers, and we have to look at the blood-brain barrier, and in the ideal world, we could identify these markers very early on, perhaps even in preclinical gene carriers. So, what are the messages? I wanted to tell you that clinical stages of ALS do exist. Perhaps we have to define them a little better in the future. Adriano and Amal Chalabi are working on that. I think we know now that systematic patterns of CNS involvement in ALS do exist, as at least shown by molecular markers. The work of the future will be, is there a clearly reliable clinical anatomical relation? And now the next if so, biomarker research should be done according to these systematic patterns. This is my contribution. More problems, but I anticipated that and I predicted it. Thank you.